Hi, my name is AHMAD FIRDAUS.
My name is NOOR.
My name is AZMAN.
My name is SITI FATIHAH. My name is Muhammad WAFIUDDIN.
Hi, in this section, we will cover about ocular embryology. Development of the eye takes place between week 3 and week 10. By the 3rd week of embryonic development, 3 primary germ layers are formed on the embryonic plate, which is ectoderm, mesoderm, and endoderm. Only ectoderm and mesoderm will take part in the developing ocular structures. It will arise retina, the iris and ciliary body epithelia, the optic nerve, the smooth muscles of the iris, and some of the vitreous humor. Surface ectoderm gives rise to the lens, the conjunctival and corneal epithelia, the eyelids, and the lacrimal apparatus, the remaining ocular structures formed from mesenchyme. Development of optic cup, and lens vesicle. As the optic surface comes in contact with the surface ectoderm, it induces it to thicken, and forms lens plate coat. So, lens is ectodermal. Development of retina. 2 layers of optics cup will differentiate into retina, which is outer layer and inner layer. Anterior portion form the iris and ciliary body, while posterior portion form the retina. Development of lens. When the retinal pigment epithelium is developing, the cells of the posterior part of the lens vesicle transform into primary lens fibers. After 4 weeks, more lens fibers are formed, from the anterior wall of the lens vesicle to primary then to secondary lens fibers. Development of choroid, sclera and cornea. In week 6th and 7th, the mesenchyme that surrounds the optic cup condenses into 2 layers, which is, an inner pigmented vascular layer which is the choroid and an outer fibrous layer which is the sclera. The mesenchyme that is anterior to the developing lens splits into 2 layers that surround the newly formed anterior chamber of the eye. The inner layer that is continuous with the choroid forms the iridopupillary membrane. The outer layer that is continuous with the sclera forms the cornea stroma. Development of iris and ciliary body. The anterior portion of the optic cup forms the epithelium of iris and the ciliary body. The non-pigmented epithelial layer derives from the inner layer of the optic cup, whereas the pigmented epithelial layer derives from the outer layer of the optic cup. The stroma of the iris and the ciliary body derives from the neural crest cells. The sphincter pupillae and dilator pupillae within the iris stroma derives from the neural ectoderm of optic cup, and the ciliary muscle however derives from the mesenchyme. Development of eyelid, and conjunctiva. At week 6, the surface ectoderm just anterior to the cornea begins as 2 folds of skin that meet over the cornea and are attached to one another. At about week 27, the 2 folds separated. How vitreous develops and forms. Development of vitreous. The vitreous forms in the center of the optic cup. The gel-like substance within vitreous derives from mesenchyme. Summary of the topic. Optic vesicle will develop first in week 3 to 4, then in week 5 to 6, optic cup, lens vesicle, choroid and fissure, hyaloid artery will develop. In week 7 to 8, cornea, anterior chamber, pupillary membrane, lens, retina, then in week 9 to 15, iris, ciliary body develop. And last eyelids will form in week 8 to 10.
In Chapter 3, we're gonna study the uvea. Basically, the uvea comprised of the iris, ciliary body, and choroid. Going further in this video, we're gonna learn it one by one. Stay calm and enjoy it. Now, let's have a look first at the iris. So, what is the iris? Iris is a thin circular structure located anterior to the lens. It divides the eyeball into the anterior chamber and posterior chamber. The iris also has central aperture called the pupil, which determines the amount of light entering the eye. Next, let us have an overview of the iris. The iris can be divided into two zones by collarette, which includes the central pupillary zone and the ciliary zone. Collarette is the thickest part, while the iris root is the thinnest part of the iris. The iris also contains two types of muscles that control the pupillary aperture, which are the sphincter and the dilator pupillae. Moving on, let us study the histological structure of the iris. The iris can be divided into four layers, which are the anterior border, the iris stroma, anterior epithelium, and posterior epithelium. The anterior border layer is a condensation of the underlying stroma. The iris stroma consists of fibroblasts, melanocyte, blood vessels, and nerves in a collagen-rich extracellular matrix. It consists of sphincter pupillae, which are innervated by parasympathetic nerves that constrict the pupil. The anterior epithelium is continuous with the outer pigmented ciliary body epithelial layer, while the posterior with the inner non-pigmented layer. Now, let's take a bit of attention to the functions of the pupil. Firstly, the pupil provides a depth of focus where the pupil constricts during accommodation. Secondly, it is a channel for aqueous flow between the posterior and anterior chamber to prevent the buildup of eye pressure. Third, it controls the optical aberrations by restricting the light into the eyes. And lastly, it broadens the luminance range for vision as pupillary changes in different background illumination. Now, what about the ciliary body? The ciliary body is continuous with the iris anteriorly and the choroid posteriorly. It is the middle portion of the uvea. Before we go further into this, let's have an overview first of the ciliary body. The ciliary body is triangular in cross-section, and it is a continuous ring inside the anterior sclera. It extends anteriorly to the scleral spur, where it is firmly attached to the sclera, and extends posteriorly but demarcated by our serrata. The ciliary body is innervated by parasympathetic, sympathetic, and sensory nerve fibers. The anatomy of the ciliary body can be divided into two parts. The anterior pars plicata and posterior pars plana. For pars plicata, the inner surface is corrugated. It also has finger-like projections called ciliary processes with fibrovascular core, surrounded by specialized ciliary double epithelium. The aqueous humor is formed over the ciliary double epithelium. For the pars plana, it is the flatter region that extends to the ora serrata. Basically, the ciliary body consists of the ciliary muscle, ciliary stroma, and ciliary epithelium. The ciliary muscle makes up the ciliary body. It has three groups of smooth muscle fiber, includes longitudinal, outer, radial, and circular, inner. Next, the ciliary stroma consists of highly vascularized, loose connective tissue. It also contains capillaries that have fenestrated endothelium, hence aqueous fluid accumulates in the stroma. Lastly, the ciliary epithelium consists of two layers, which are the outer pigmented epithelium and inner non-pigmented epithelium. The epithelial cells in these two layers are arranged apex to apex, forming gap junctions that allow rapid solutes exchange. Tight junctions around the margin of the non-pigmented epithelium, NPE, cells form the major blood aqueous barrier. So, what are the functions of the ciliary body? Firstly, the ciliary body produces aqueous humor. Secondly, it involves accommodation via contraction of the ciliary muscle.
Now, let's learn and know about the aqueous fluid. The aqueous fluid is a clear plasma-derived unfiltrate, which means no blood encounter this fluid. The formation of aqueous fluid is mainly by diffusion, ultrafiltration, and active secretion. Simple diffusion is caused by osmotic pressure gradient between ciliary stroma and capillaries. Only small volume are forced out capillaries. Ultrafiltration are caused by hydrostatic pressure that affected by the gravity force. Plasma is pushed through fenestrated capillaries, creating unfiltrate within ciliary stroma. In active secretion, it is a energy dependence that accounts for 80 to 90% of aqueous production. Secretion occurs at the ciliary epithelium. Once aqueous fluid secreted into the posterior chamber, the aqueous fluid passes through the pupil into the anterior chamber for exit. 1. Trabecular meshwork route. The aqueous passed through the trabecular meshwork and across the Schlemm's canal, then the aqueous passes into collector channels, aqueous veins and into the episcleral veins. Trabecular meshwork route accounts for majority of aqueous drainage. Second pathway is uveoscleral route. The aqueous passes the anterior chamber angle into the connective tissue space within ciliary muscle via iris root and ciliary body. Function of the aqueous fluid are deliver oxygen and nutrients. 2. Remove waste product from inflammatory processes. 3. Remove other cellular debris from posterior cornea and crystalline lens. 4. Provision of low refractive index transparent medium between the cornea and lens. 5. Maintain the intraocular pressure for optimal shape of the eye. Let's move to the posterior portion of the uvea, which is choroid. Characteristics of the choroid is: 1. Choroid extends from oris or rhoda to optic nerve. 2. Choroid are highly vascularized layer between retina and sclera. 3. Choroid attachment is firmest at the posterior pole. 4. Choroid is thicker around posterior pole than anteriorly. This is histology of choroidal layer. Starting from the top, layers of sclera cover the choroid, following by lamina fusca or suprachoroid lamina, then stroma layer, which pigmented layer, choriocapillaries, that provides nourishment to the outer retina. Holler's layer is a larger vessels and Sattler's is a medium-sized vessel found in stroma. The innermost layer is basal lamina or Bruch's membrane, and retinal pigment epithelium. The main function of choroid is provide nutrients to the outer retina. 2. Choroid absorbs excess light that passed through retinal pigmented epithelium layer. 3. Suprachoroidal space provides pathway for posterior vessels and nerves that supply the anterior segment.